Okay, so let's go ahead and move on from talking about the peripheral nervous system and move on to the central nervous system here just for a brief little bit. Now, the central nervous system, really the main functions of this is going to be to collect the information that's being provided by the peripheral nervous system, to integrate that information together in some way in order to make some decisions about that, and then formulate responses and transmit that information back out to the peripheral nervous system again. Now, if we're talking a little bit about what actually makes up the central nervous system, there are several different ways that we can talk about. First, we can talk about the organs specifically that are making up the central nervous system, and that's going to be the brain and the spinal cord, which we'll talk a little bit more later. But we can also talk a little bit about the types of cells that are making up the central nervous system as well. So in terms of neurons, all interneurons are going to be part of the central nervous system. Many of the efferent neuron bodies, I'm not talking necessarily about the axons, although part of the axons as well, but many of the efferent neuron bodies are going to be part of the central nervous system as well. And then also many of the axon terminals of the afferent neurons are also going to be part of this central nervous system. So that's kind of the parts of the neurons that are making up the central nervous system. And then this other cell type that we haven't at all talked yet about are glial cells. So what in the world are glial cells? Well, it actually turns out that about 90% of all the cells in the human central nervous system are glial cells. So most of what's making up your brain and spinal cord are actually not neurons at all, they're glial cells. And what it turns out is if you look across the animal kingdom, the larger and more complex brains and central nervous systems get, the higher proportion of glial cells make up those brains. So if you look at something with a much more simple and small central nervous system, you're going to have many, many fewer glial cells and up to us. And actually there are a few things that actually exceed us. I think, for instance, whales have somewhere up around 95, 98% of their cells, their central nervous system cells are glial cells. So there's several different functions that these make up, but really what glial cells are there for is to help structural and functional support of the central nervous system. And there are four major types of glial cells that we'll be talking about here. First off, one that we've talked about already, the oligodendrocytes, also astrocytes, microglia, and ependymal cells. Let's talk about these each in order. So first off, we visited oligodendrocytes before, and their major function is going to be to create the myelin sheath in the central nervous system. So these are doing essentially the same function that Schwann cells were doing out in the peripheral nervous system. And there's a few other functions, but nothing major that we really want to talk about here. This is a micrograph of a oligodendrocyte, and there's been several proteins that are specific to myelin sheaths that have been stained in this, and what you can see here, you can't see it very well, but there's a neuron that comes through here, so you can kind of see the outline of the neuron that this oligodendrocyte is sheathing, and there's another neuron here that's being sheathed, and so on. So just kind of give you a, what does this actually look like sort of thing, oligodendrocytes. Okay, now there's also astrocytes. Astrocytes are so named because they kind of look like stars, astro, so. And astrocytes really are going to be kind of one of the jack of all trades of the central nervous system. These cells are going to be doing a ton of stuff to support and help the functioning of the nervous system and do really a ton of stuff. So one of the major things that they do is they help form the structure of the central nervous system. So these are actually providing a scaffold that helps hold neurons in place into the correct place. And then they also form kind of a glue that also holds it together. So astrocytes play a huge role in the structure of the central nervous system. They're also going to be transferring a lot of nutrients to neurons. So there's actually a lot of metabolic processes in the neurons that astrocytes are helping to support. They'll be picking up a lot of nutrients out of the blood and then passing them on to the neurons as well. Not all of them, but there's a good number of them. So there's a lot of metabolic interchange between astrocytes and neurons. Also, astrocytes are going to a small amount of glycogen, and this is this really short store of glucose for neurons. Now, neurons are absolutely dependent on glucose as an energy source. A lot of the rest of your body is not. They can survive on a few other things. So really neurons need glucose, and maybe in a pinch can use ketone bodies as well as a fuel source. But two things that neurons really need, glucose and oxygen as well. Without either of these for even just a few minutes, neurons begin to die very quickly. Now, these astrocytes store a small amount of glycogen, which we'll talk about later when we get to metabolic physiology, is a storage type or storage molecule for glucose. It can be broken down to glucose very readily and can provide some limited amount of glucose for neurons, but really it's not much at all. Also, astrocytes help in repair the central nervous system. Now, by repair, we don't necessarily mean reconnecting neurons. If neurons have been damaged, oftentimes astrocytes will move in there and help form scar tissue and help fill the dead space. Now, they're not necessarily going to put the neurons back together, but by filling this dead space and forming like this astrocyte scar tissue, they can help somewhat in the repair of central nervous system damage. They also take up used neurotransmitter and also sometimes release neurotransmitter, both in a communication role, but then also just supplying neurotransmitter to neurons. So one thing I mentioned a while back about how does neurotransmitter actually get out of the cleft, and one of the ways is they'll simply diffuse out of the synaptic cleft. Well, what happens to it after it diffuses out of the synaptic cleft? And the central nervous system, one of the major things is that astrocytes will actually take up a lot of that used neurotransmitter and begin to recycle it. Also, astrocytes play this really big role in regulating the composition of the brain extracellular fluid. So the extracellular fluid, because neurons need to have a very specific environment to maintain the electrical activity, they need to have extracellular fluid of the brain being held in very, very tight margins. And so astrocytes are really going to help regulate that kind of composition, and particularly they're going to be helping in regulating the concentrations of potassium in the extracellular fluid. Because remember, we need to have potassium flooding out of the cell as we reset our action potentials, bring it back down to resting potential. And if we have too high concentrations of potassium outside the cell, and we're diminishing that concentration gradient, we can start having problems generating and propagating action potentials, and graded potentials for that matter. So this is an important role of astrocytes. Then also they help in synapse formation. So we're talking a little bit earlier about how astrocytes help form structure of the central nervous system. And one of the things that they do is they actually in some ways, hold neurons apart from each other by projections that they're putting in between these neurons. Now, astrocytes can actually then, in closely spatially oriented neurons, can actually form projections in between them to keep them physically separated, but then also withdraw those projections and allow a synapse to form between neurons, and this way kind of regulate synapse formation within neurons, so they can help play a larger role in new synapse formation. So again, just to kind of show you what's going on with astrocytes, what do they look like? In the lower right-hand corner here's a green fluorescent protein stained astrocyte, and you can see you have these many projections that are kind of weaving their way out into the surrounding tissue. So kind of what an astrocyte looks like. Next thing that we're going to talk about is ependymal cells. Now, ependymal cells are kind of interesting. They line the internal cavities of the central nervous system. So you have these cavities like ventricles or openings inside your central nervous system, like your brain. They're going to allow cerebral spinal fluid to flow through them. And ependymal cells are going to be lining these internal cavities. And it turns out, as they're sitting there, they're helping contribute to the formation of cerebral spinal fluid, which cerebral spinal fluid is just the fluid that is surrounding the brain, is inside the ventricles of the brain, also surrounding the spinal cord as well. Now, this isn't exactly the same as the extracellular fluid of the brain, but the chemical composition of the cerebral spinal fluid is going to have a large impact on the composition of the extracellular fluid. So it's important that we regulate the chemical composition of the CFS quite a bit as well. Also, ependymal cells have these cilia, that allow them to help circulate the cerebral spinal fluid throughout these ventricles, throughout these open spaces in the central nervous system. And then also, this really other interesting activity of the ependymal cells is that they serve as kind of stem cells for the other nervous system cells. So, for instance, glial cells, ependymal cells can become, and the other three types of glial cells, actually, excuse me, I believe they can only become astrocytes or oligodendrocytes, not microglia. But they can become these other types of glial cells. Also, they can become neurons. Now, it's kind of been this idea that you don't form new neurons in the central nervous system. Well, it actually turns out that you do. Ependymal cells can become new neurons in a couple specific places, specifically the hippocampus and also the subventricular zone. This layer below the ventricles in the brain also has some creation of new nervous cell or neurons as well there. So ependymal cells can serve in that function. Kind of looking here at a cross-section of the brain, here lining this cavity here, these are all ependymal cells. You can kind of see the change in the type of cells you get there. And in some of these, I don't know, no, you can't really see the cilia here, but that's kind of what ependymal cells look like. And then the last glia or glial cells here, we're going to talk about microglia. These are immune cells of the central nervous system. They're actually descended from the same cell lines that monocytes are. And they're going to be scavenging debris and other foreign objects in the central nervous system and really providing the one of the main lines of immune defense once you're inside the central nervous system. So those are the glial cells. And here we get some, in the green here, these are microglia here. Both the round ones and the kind of wispy ones here, and the red all throughout here are neurons. So the microglia actually tend to be somewhat wispy and branchy under normal conditions in the central nervous system, but once there is something that they need to respond to, they tend to ball up, like you're seeing over here on the left, and then go do what they're going to do. Okay. So now we talked about the different cell types. Let's talk about the kind of overall organization of the central nervous system. There's a couple different ways that we can talk about. But first, we can talk about the type of matter that's making up the central nervous system. And the first one that we're going to talk about there is gray matter. So gray matter is, if you look at a slice of nervous tissue, it looks literally gray. And the reason for that is this is being made up of neuron cell bodies and dendrites, and also of glial cells. And so this is the area that a lot of the information processing is going on, because a lot of the synapses between different neurons are happening here in the gray matter. So a lot of that information exchange is happening here. Then we also have white matter, which is made up of the myelinated axons of neurons. So it actually turns out that myelin, being very lipid rich, appears white, and so making the white matter of the central nervous system appear white. And really the white matter is going to be primarily involved in the conduction of information from one place to another. That's just kind of overview of what's going on there. So let's take a look at the organization of the different organs. First we have the spinal cord that runs from the base of the skull all the way down to the sacrum, which is kind of the dorsal part of your pelvis. And this is a cross-section here of the spinal cord, and just kind of orient you. So this over here is the dorsal side on the top here. The dorsal side being towards your back. And then down here is the ventral side of that. And what you'll see is we're organized into this inner medulla, essentially, of gray matter. So this is going to be some information processing happening here. This is mostly made up of cell bodies and dendrites. And then outside here, kind of surrounding the gray matter, we have this white matter. So this is mainly neuron axons, conducting information both up and down through the spinal cord. Now let's talk about the gray matter. The gray matter is actually kind of divided up into these three different sections. So the first section is right over here. This is the dorsal horn of the gray matter. Then we have this kind of little protrusion right here. This is the lateral horn of gray matter. And then down here, this large section right here is the ventral horn. I'll come back and talk a little bit more of what's going on here in just a few moments. But then also you have this area right here. This is the central cavity. And there's this, it's just a small cavity that you actually have cerebral spinal fluid flowing through that. And so that's kind of the general organization of the gray matter. So these dorsal horns are both left and right. They're paired. So the dorsal, lateral, and ventral horns are all paired through there. So let's talk a little bit about the white matter. So how is the white matter arranged? It actually turns out that the white matter is arranged into these tracts of axons that are going to and coming to similar places, so essentially the same places. And they're named based on where they're coming to and where they're going to. Okay, so for instance, if we look over here, we have, for instance, the spinocerebellar tract. Or let me back up here. Not only do we have where they're going to or coming from, they're also divided up by efferent and afferent pathways. So if you look at all the red, these are all efferent pathways. So these are going to be descending pathways, or pathways that are going from the brain or somewhere higher in the spinal cord down to somewhere where they're going to leave the spinal cord and be efferent out into the body. We also have the sensory or afferent pathways that are going to be coming from afferent neurons that are going to then synapse somewhere here in the gray matter, and then they're going to go ahead and come out, synapse to some sort of interneuron that's going to come here and then ascend either somewhere else in the spinal cord or up to the brain, which is most likely. So let's come back here. So for instance, we have afferents and right here, the spinocerebellar tract. So this is coming from the spine going to the cerebellum. Or we have the spinothalamic tract, which is coming from the spine going towards the thalamus. And we also have other ones. For instance, we have the cortical spinal tract, which is coming from the cerebral cortex, going down the spinal tract, and going to these different places. For instance, exiting in the sacrum, the lumbar, and the thoracic or cervical areas. So, just generally known that we have these spinal cord tracts that are axons grouped together by where they're coming from or going to, whether or not they're efferent or afferent pathways, and then also where they're going. And so we're keeping information going to spatially separate places, spatially segregated in our spinal cord as well. Also, what we have is we have spinal nerves that are coming to and leaving the spinal cord as well. And so this is kind of showing you what's going on there. We have our spinal cord here, kind of off to the right-hand side. And then we have the spinal nerve leaving there. And there's a couple different parts here that you need to know. There is both the dorsal root and the ventral root here. So the spinal nerve is going to be leaving in two different places. And it's actually, we'll talk a little bit more about why they're different, but one thing that you'll notice is both of these roots are essentially departing from one of these horns. And so these gray matter horns are essentially extending towards the spinal cord or the spinal nerve roots. One other thing that we need to talk about here is this little bulge right here along the dorsal root, and what this is the dorsal root ganglion. So the dorsal root ganglion is this collection of cell bodies that is going to be located outside. These are mostly afferent cell bodies, or exclusively afferent cell bodies. So let's kind of take a look real quick about how our neurons going to be coming through here. And if you look here, this blue side, this blue neuron is an afferent neuron. You can tell you have your cell body kind of off to the side here. These green ones are interneurons. And then the red one here is this efferent neuron. And so the ventral root is generally taking these afferent neurons, and your efferent neurons are traveling through the dorsal root. Dorsal root is accommodating these afferent neurons, whereas the ventral root is accommodating these efferent neurons. So you'd have an afferent neuron coming in this way. The cell bodies are generally going to be located here in this dorsal root ganglion. Going to move inside where it can synapse with some interneuron. And these interneurons are going to be in this gray matter. And then oftentimes completing a reflex loop, they'll be synapsed onto an efferent neuron that's then exiting through the ventral nerve root. Or they could synapse onto another interneuron that will then move out, or its axon will move out into the white matter and then ascend towards the brain, carrying this whatever sensory information is coming from that neuron towards the brain. So that's kind of how things are organized here in the spinal cord. Okay, so let's move on from the spinal cord and talk very, very briefly and very, very broadly about the brain organization. I'm not going to really go into much detail here, and really, we could take an entire class just talking about the physiology and the processing that's going on in the brain. It's a very, very, very complex organ, and I wish I had more time to talk about it. I wish I had a quarter or a semester or a year-long class that I could just spend several weeks talking about the brain, because there's really a lot of amazing stuff going on here. However, to get everything in, I'm just going to briefly talk about this. We're going to come back to it actually fairly often and talk about it, how processing of the brain is going to be impacting other physiological functions. So really, right now, just for the moment, I'm going to just be giving you the basic ideas or the basic locations of where generally the gross anatomy of the brain is, and we're going to leave it there, because we're going to come back and talk more about this in pieces as we talk about other parts of the physiology. So, really the brain is organized grossly into three different parts. We have the brainstem, the cerebellum, and the forebrain. So, let's take a look. So, this green area right here is the main parts of the cerebellum, or excuse me, the brainstem. So what we have down here is the medulla. This large kind of expanse right here is this other area called the pons. And then really we have the midbrain, which isn't colored in green, but would really be all the way through here, which is also part of the brainstem. So I'm going to call that, up here, the midbrain. And the brainstem is going to be taking care of a lot of kind of processing autonomic nervous system inputs and stuff like that. So a lot of stuff that's not going to rise to conscious level, and also a lot of stuff that just controls the basic homeostatic function of the body. It's going to be taken care of here. So that's basically what's happening in the brainstem. Moving on, we also have right here the cerebellum. And the cerebellum is going to be taking care of a lot of the processing that goes into body positioning. So balance, posture, how are we just keeping ourselves in kind of fine moving condition autonomically from a subconscious level. So that's what the cerebellum is doing. Now also we have the forebrain. So the forebrain is really the rest of what's going on here. We have the cerebrum here, which is the main, when you think of a brain, and you think of all the kind of the folded main part of the brain, you're thinking of the cerebrum. And the outer part of this, unlike the spinal cord where the gray matter was in the middle, and the white matter was along the edges, what we have going on in the brain is that most of this gray matter now is going to be on the outside in the cortex, and the white matter is going to be in on the inside. And so that gray matter along the cortex, we're going to call that the cerebral cortex. And this is where most of the conscious processing is happening. It's up here in the cerebral cortex. But there's also a lot of other processing that's going on there. This is kind of the main workhorse of processing of the brain. Another one that I just really want to talk about here is there's this organ that's situated right kind of here that's hard to see. Sitting here like this, called the thalamus. This is going to be a major relay center for afferent information. It's going to be going to different parts of the brain, especially up in the cerebral cortex. So a lot of our special senses end up being transferred to different parts of the cerebral cortex for processing. And that's most of that relay is going to be happening in the thalamus. And down here is the hypothalamus. Down there. So really, I'm not going to talk a whole heck of a lot about all of this. There's more parts that we're going to add on later on. But really, this is kind of the gross anatomy of the brain. And we're just going to leave it there, as much as I hate to do that. But we'll come back to this again a little bit here and there as we're talking more about physiology over on. Come back talking more about the brain. So really, this is going to kind of finish up our talking about the the nervous system proper, like the functioning of the nervous system by itself. We're going to move on a little bit now to sensory physiology, and then move on from there to be talking about other things. But really, the nervous system and what we've talked about so far is going to be coming up again and again and again, because it's doing so much to regulate different areas of physiology.